Hello, everyone. My name is Nathan Rather, and I'm an undergraduate student in the Gauche Lab. And today I'm going to be sharing with you my work there. So the Gauche Lab is generally very interested in the elimination of so-called morphologically complex cells. These cells have vast architectures and different subcellular compartments that extend into different microenvironments. And we ask how these cells with such complex architectures are eliminated. And in order to study this, we use the nematode C. elegans because it's an ideal model system, and its cell death pathway is genetically conserved across metazoans. And the cell death pathway proceeds with EGL1 repression of SED9, which represses SED4, which is the main activator of the executioner caspase SED3. And we use the novel cell death program called compartmentalized cell elimination, or CCE. One place in which this occurs is the tail spike epithelial cell. And CCE proceeds where in three different parts of the cell, the soma, the proximal process, and the distal process die in three separate ways. And importantly, CCE is a non-canonical apoptotic cell death program because it occurs independently of EGL1 and it's weakly dependent on SED9. And in order to gain better insight into the genetics of CCE, we performed a number of forward genetic screens. And one forward genetic screen mutant called NS957 shows a persisting soma compartment remnant in L1 larva, wherein the proximal process had been removed. And keep in mind that CCE occurs in the embryo, and so any sort of tail spike cell in the larva is highly abnormal. And I found that the causative lesion of NS957 is in a gene called EOR1. Multiple different loss of function EOR1 alleles phenocopied NS957's defect, and EOR1 reintroduced into NS957 rescued the defect, and EOR1 is also expressed in the tail spike cell. And a number of binding genetic partners for EOR1, EOR2, and a number of chromatin modifiers, loss of function alleles for these genes also have a persisting soma defect. One of the interesting things about the EOR1 persisting soma is that it's rounded, enlarged, and has these persisting visible nuclei. And it doesn't look like a dead but unengulfed tail spike cell. And it also doesn't look like a living tail spike cell. And a double mutant confers this, suggesting that this EOR1 mutant is sort of neither living nor dead, and also that EOR1 may act to promote the killing function of the tail spike cell. It doesn't specify its death, and it also doesn't engulf the cell. And we wanted to know where in the genetic pathway of CCE, sorry, it's over here, where in the genetic pathway of CCE does EOR1 exert its effect? To do this, we used a reporter for caspase activity, and we found that caspase activity is normal in EOR1 mutants, suggesting that EOR1 may act downstream of SED3 caspase. And a double mutant with SED4 confers these results. And so we wanted to know what the gene could be that EOR1 is influencing the expression of, given that it's a transcription factor. So it modifies gene expression. And so we looked at a list of EOR1 genetic targets, and one gene stood out to us called WA1. And WA1 is this mitochondrial protein that's important for mitochondrial function. And upon activation by SED3 caspase, it translocates to the nucleus to degrade DNA. And a loss of function allele for WA1 phenocopies EOR1 persisting soma defect, so it looks like EOR1. And a double mutant between EOR1 and WA1 does not show a synergistic increase in tail spike cell phenotype, suggesting that EOR1 and WA1 act in the same genetic pathway to promote soma elimination during CCE. And so given that that WA1 is a mitochondrial protein, I wanted to know what mitochondria looked like in these persisting soma remnants. And I found that they actually looked very distinct from SED3. SED3 had these sort of normal reticular mitochondria, these sort of normally shaped in this living tail spike cell mutant. And in the EOR1 and WA1 mutants, they appear sort of degraded. So this really suggests that something's happening at the mitochondria. And I next want to know what the nature of these mitochondria are. Are they fragmented? Are they being degraded? What's going on there? And so overall, I think that EOR1 is controlling the transcription of WA1, which is this important mitochondrial protein that is being activated by SED3, where it degrades DNA. And the loss of WA1 is what leads to this soma phenotype.